on my day. Cold as hell, man. Cold as shit. On to amino acids, precursors. Ions are cyclic. Viola. Rudderless form. There was intermediate video. It's liver. They're producing solid with bone marrow. The liver heme is required for the cytochrome B. In erythroid cells, heme synthesis, iron, globin synthesis. Liver and erythroids. Synthesis told you of the cytochrome P450 and they produce. Globin synthesis. Driven by mitochondria and cytokine. Lysine section on different. Alright, we've got delta monolithic. All the carbon and nitrogen. Two glycine, 2,6-alcohol. Adenosine, hydroxyl group over. Adenosine. Four of these condense for hydroxymethylylane. Group over adenosine. Four. Yo, what is going on? It's not too bad, but it's 5, 6. Hi. Glycines are gonna go ALA synthase. Final adenosine dehydrated. Carbon, nitrogen atoms. Condense mitochondria. PLP dependent. Vitamin B6. Rate limiting step. How much you done with? I've got biochem left and anatomy, all of biochem. I'm gonna finish probably like most of biochem today, and then tomorrow the rest of biochem and anatomy. No embryo, I'm dipping on embryo. I'm just gonna chat GPT it. Like barely. I'll give like 2 hours to embryo max. Not looking good. I don't know, bro. I mean, I only did one chapter of biochem. This is the second one. This isn't too hard, to be honest. I'm gonna be done with this. It's 20 minutes. 20 minutes, I'll be done with this shit. And then I'll head on to glycolysis. Old as hell, man. What do you have left, by the way? You know, 1 to 8 physio, got blood left, entire anatomy, entire biochem left. I don't think doing embryo is a wise choice. Block 1, I dipped on all of embryo and all of histo, and I got 17 out of 28. Nothing crazy, but like, it's possible. In anatomy. Paradoxal phosphate dependent. Omitted rate limiting step. Isoforms ALS1, ALS2. ALS1 is hepatic. Housekeeping erythroid specific. Nutrition depends on iron availability. When heme synthesis exceeds demand. The monitor's here? Why? I thought 4K was like a massive bump or some shit. How are you feeling about your financial decisions? Brightness ज़्यादा है. Was that a wise choice? Yeah, you're much better than me. 18% up. My financial decisions are horrible, all based upon intrusive thoughts. Oh yeah. Is gold still a good investment? I mean, I feel like gold is always the best investment, is it? I mean, that's what I just hear. Gold काफी महंगा हो गया. Back when I was a kid, it wasn't as expensive as it is now. hedge against inflation. Oh. Oh yeah, makes sense. So it seems like it's worth more. Purchasing power is all the same. Okay. So when should someone invest in gold? Diversify. Do you think I make enough to invest in stuff? Because I personally make around 50k a month. Depends on the month. 40 to 50k. More than enough? For 30 years comes 15 crore. God damn. I know I asked you this a while ago. You told me to invest in mutual funds back then. Have a 6-month emergency fund. That's what I'm working on still. Get that 6-month emergency fund alongside my expensive. And you save 10k. Yeah. That's a good way to think about it. Damn, bro. 1.5. Okay, I gotta fix my habit, man. I spend too much money on bullshit. .5%. I need more net worth to actually. Reward system, man. Ah. But then how do I calculate my net worth? How do I know what my net worth is? On one spreadsheet. मैं हर मंथ में 3-4 बार क्लोजिंग करता हूँ अपनी पर्सनल नेटवर्थ. You're saying 0.5% of that is what you allow yourself to spend on luxuries. And what do luxuries include for you? Kapde shapde, bahar khana, that type of shit. Do not need but want. Okay, there's a ton of that. It's not real luxury. I spend a lot on takeout food. Yeah. Okay, I don't have much food problems. I just cook myself. Or when my mom's around, she makes it for me. She's around these days, so I'm chilling. Thing is, I'm like in a pretty weird financial situation. Like I have more than enough, but I still want more. But like I don't need more. So that kind of like demotivates me. So I don't know what to do, to be honest. So that's why I just keep on spending money on dumb shit. But then I want more. I don't know, man. It's so stupid. That's everyone? I mean, yeah, fair enough. I do have huge ass goals, but those come after I freaking graduate from this place. Nah, fuck the Chinese cars. Nicaragua can not be the bank. Ah, you mean it that way? No, definitely no. I don't think I can. I mean, I'd have to sell something, but it wouldn't be like a smart thing. It would be kind of retarded. वो निकाल के फिर मैं सड़कों पे आ जाऊंगा, गाड़ी में रहूंगा. करोड़ की गाड़ी है. हाँ. I'm just thinking about my assets. Wait, what do I have? That is the goal. Yes, you're right. Shit, man, you got me thinking. Anything appreciating over time. Appreciate कोई पता नहीं आजकल real estate के बड़ा shit हालात है. I've only got real estate, and that depreciated a lot, like much lower than what it was supposed to be. Yeah, fucking dog shit, man. Used to be good. Established. We should. I also used to tell my parents invest in commercial and shit back when I was younger, but they never listened to me. I wish they did. Dolmen, Mallman, rush karo. Okay, let me keep this on the side one sec. Without the hassle. What's the hassle? Okay, the hassle. Okay. You talking about this? DCR? Dolphin City rate. The thing is, okay, I invest, but I have zero idea about any of what this shit means. I have never ever looked into stocks. See that the payout 255 per year per share. Here. What's EPS? Or do you sell some courses too? I don't know how to figure out any of this shit. Yes, definitely. Definitely. As soon as I'm done with this bullshit exams. All the same info on the paid and unpaid courses, yeah. I mean, it's just how people make money with the paid courses, right? Would you tell me to invest right here, right now, on this? And if I was supposed to, how am I supposed to do it? And how much should I invest in this? 20 minute vids. Oh, these are pretty short.
अगर मैं आपको कहूं कि स्टॉक मार्केट में इन्वेस्टमेंट करना
Okay. Basket of stocks. Okay. What's MII then? ESX.com. Search good man. MII ETF. Okay. Should I go all in on this? Click underlying basket per 10,000 ETF units. ETF. Underlying basket per 10,000. Okay. Alright, I found it. Once you get 10k ETF shares, this is how it's divided. Oh, so it invests in different companies for you? Is that what it is? Polyfax, no. Skin ointment, acha. Polyfax. Comex and B-sulfate. Okay. Different companies for you. Right? It's okay, Polyfax go. What about it? Anybody? Ever seen Polyfax? Made by GlaxoSmithKline, GSK. And is GSK one of these? GSK. Most used screen admin in PK. Really? Cornflakes? हाँ cornflakes का तो पता है यार. Achha, Fauji, yeah. Bro, Fauji ones are dog shit though. Personally, I don't like him. But what about him? Who makes him? Fauji Foods. Sasgari equals rickshaw makers plus Helva H6 makers. What on earth? How? Oh, this one, Sazgar. ये वाला फौजी, अच्छा ये रहा फौजी, इसके अंदर 61 शेयर, एक शेयर. तो मतलब 10,000, ये कितना होगा? ये तो लाख कुछ होगा ना, 1,60,000. एक साठ में वो सब कुछ मिलेगा. 160 कुछ, 160 2,100. And then what about this? Is this something I hold on to? Glycine succinylcholine को ALA बनाते हैं. दो enzymes हैं. ALA synthase 1, 2. 1 is located in the liver, 2 is located inside of the erythroid tissues. Acha, both of these get inhibited in heme synthesis. If he's demand, heme is oxidized to hemin. Okay. Paradoxal phosphate. Mid-step glycine succinylcholine A. Sure. When heme synthesis exceeds demand, heme is oxidized to hemin, which feeds back on hemin, which feeds on ALS1. Hemin decreases ALS1 by reducing ALS1 gene transcription, destabilizing its mRNA, inhibiting mitochondrial import of the enzyme. This maintains heme homeostasis and prevents it. Okay. So this ALS1, it's inhibited. There's like end product inhibition, and ALS1 is found inside of the liver by heme itself. Heme, what it does, it reduces the transcription of ALS1, it messes with its mRNAs, and it prevents a lot of it from entering inside of the mitochondria where this first reaction takes place. Say hemin regular. Hemin is oxidized hemin with ALS. The hemin is oxidized hemin. Intracellular iron. ALS2, ठीक है. Intracellular iron ज़्यादा होगा तो barbiturate, antibiotic, P4. Is heme dependence with heme, lowering the hepatic heme pool, used heme, removes repression, increase ALS, enhance the ALS profile. Lowering the formation of porphobilinogen, ALA dehydratase, lead inhibitor, zinc dependent. In the site. Similarly condensed to form porphobilinogen. Catalyzed by ALA dehydratase. Zinc enzyme. Yes. porphobilinogen, uroporphyrinogen. Before uroporphyrinogen is formed, porphobilinogen, porphyrinogen, porphobilinogen. What the hell are these names, bro? Hydroxymethylbalane. Four molecular porphyrinase condense to form hydroxymethylbalane, which is then cyclized and isomerized by uroporphyrinogen 3 synthase to uroporphyrinogen 3. Intermediate decarboxylated by uroporphyrinogen decarboxylase to form coproporphyrinogen. Okay, 4 of the porphobilinogen synthases, porphobilinogen hydroxymethylbalane synthase, hydroxymethylbalane synthase. Both these condense to form it, and then it gets cyclized and isomerized by uroporphyrinogen 3 synthase to form uroporphyrinogen 3, which then gets decarboxylated to form coproporphyrinogen 3. All of these occur inside of the site as well. Version of heme. Coproporphyrinogen 3 re-enters the mitochondria. Protoporphyrin 9 and then protoporphyrin 9. Protoporphyrin 9. Pericletase. Synthase, uroporphyrinogen 3 synthase, uroporphyrinogen 3 decarboxylase, coproporphyrinogen enters the mitochondria, decarboxylase again, form protoporphyrinogen 9, which is then oxidized to protoporphyrin 9. Pericletase catalyzes iron. Insertion of iron to protoporphyrin. Form heme, finally. LED hydratase inhibited by lead. Yes. Alright, I like that. Pretty easy.
Hello everyone, I am the student of Dr. Adil Bandu, Professor of Anatomy and Neuroscience, Mansoura University, Egypt. I have heard that Block B paper was very tough and that my lectures were not enough for that. Since many of you prepared from slides, I had provided you first difficult time and I am really ashamed of that. Since you people
Porphyrias. Let's just do the whole thing once more. You got two succinyl-CoA and glycine. They condense to form delta aminolevulinic acid. This is by two enzymes, ALA synthase 1 and 2. ALA synthase 1 is found in hepatic tissues. ALA 2 is found in erythroid tissues. ALA 1 is inhibited by hemin, which is an excess form. Yeah, and ALA 2 is inhibited by iron, iron. And all of this enzyme also requires pyridoxal phosphate. Now, this amino levelic acid then leaves the mitochondria and goes to the cytosol, and it is then two amino levelic acid condense to form orphobolinogen. This is ALA dehydratase, and it is inhibited by lead. Orphobolinogen is then, four orphobolinogens are condensed to form hydroxymethylbilane by hydroxymethylbilane synthase, and then this is then cyclized and isomerized to form uroporphyrinogen 3 by uroporphyrinogen 3 synthase. Uroporphyrinogen 3 is then decarboxylated by uroporphyrinogen 3 decarboxylase to form coproporphyrinogen 3. This then moves back into the mitochondria, and then it forms protoporphyrinogen 9 and then protoporphyrin 9. Then ferrocletase comes up, and protoporphyrin inhibited by lead. Addition of iron forms heme. Finally, we're done with this shit. Now, what are porphyrias? Porphyrias are a group of rare metabolic disorders caused by deficiencies of specific enzymes. Rare metabolic disorders caused by deficiency in specific enzymes that heme biosynthetic pathway, resulting in accumulation of porphyrins or their precursors in tissues and increase in excretion in urine or feces. And increased excretion in urine or feces. In tissues and increased excretion in urine or feces. Most porphyrias are inherited as autosomal dominant conditions, except for a few rare recessive forms. Depending upon the unique location, pattern or intermediate, depending on the enzymatic block. The disorders are classified by primary site of enzyme deficiency. Hepatic porphyrias, enzyme defect in liver. Erythropoietic porphyria, enzyme defect in bone marrow. Erythropoietic porphyrias, hepatic porphyrias. Clinically, porphyrias manifest as acute neurovisceral attacks or acute cutaneous fluid. The former due to accumulation of early precursors, and the latter due to buildup of later porphyrin intermediates. Comes from the Greek purple to the red purple. Acute, chronic, acute, neurophysic, abdominal symptoms, mainly cutaneous photosensitivity. Patients with defect before tetrapallar formation, and with abdominal pain. Present with abdominal pain, neuropsychiatric disturbances, and autonomic dysfunction. Abdominal neuropsychiatric disturbance. After tetrapallar, photosensitivity and itching, blistering on light. Porphyria acute tarda. Most common porphyria. The one common, it is chronic hepatic disease caused by deficiency of uroporphyrin and decarboxylase. Clinical expression include factors such as hepatic iron overload, alcohol use, estrogen therapy, hepatitis B or C infection, HIV infection, hepatitis, alcohol use, hepatic overload. Middle age with photosensitive skin lesions, blistering, hyperpigmentation, sun exposure, urine appears red, brown, and normal light, and acute hepatic porphyria. The acute hepatic porphyrias include rarely dehydrated, acute intermittent, rarely dehydrated deficiency, acute intermittent porphyria. Hereditary porphyria. With acute attacks of abdominal pain, vomiting, motor weakness, and neuropsychiatric symptoms. Unlike PCAT, may or may not. 35 include drugs, barbiturates. Hereditary porphyria. Erythropoietic porphyrias. But two types. Thread cells. Severe photosensitivity. Erythropoietic porphyria is caused by enzyme deficiencies in erythroid cells of bone marrow. Present congenital erythropoietic Ginther's disease. Congenital erythropoietic porphyria or erythropoietic porphyria. Congenital erythropoietic porphyria. Directly. Before decreasing production in the liver, removes the normal feedback inhibition on it. One, the increase in the surveillance and versus acute intermittent porphyria. Acute intermittent porphyria. The only one. Acute intermittent porphyria. Oh, still going, yeah, bro. That's how you gotta study. You know what I'm saying? Type shit. Acute intermittent porphyria. 6 hours? Are you crazy? My days, bro. And then we got hydroxymethyl, ibuprofen synthase, congenital erythropoietic porphyria, decarboxylase porphyria kidney tarda, copper porphyrin oxidase is hereditary copper porphyria. Very good. Copper porphyria. Erythropoietic protoporphyria, ferrocletase. Alright, man, alright. We get it. We get it, buddy. Reticuloendothelial system of the liver, spleen, and bone marrow. 80% of human mutation originates from senescent or breast turnover, immature RBC, and other heme proteins. Bilirubin, yellow pigment excreted into bile. Conserves iron. Carbon monoxide signaling molecule. Toxic structure. Visit that later. Formation of bilirubin. Heme oxygenase. Catalyzes the first and rate limiting step of heme breakdown. Heme oxygenase, NADPH, and oxygen. Releases hemin, Fe3 plus, and carbon monoxide. NADPH plus. Biliverdin. Biliverdin is then reduced by biliverdin reductase to bilirubin, red-orange delivered in hemoxygenase. O2, NADPH, got carbon monoxide, Fe3 plus. NADPH again, biliverdin reductase, bilirubin formation, hemoxygenase bilirubin phages in liver, spleen, and bone marrow, system, bilirubin, unconjugated bilirubin, hydrophobic, lipid soluble. Is insoluble in plasma, so travels non-covalently bound to albumin in the liver, while within hepatocytes it dissociates from albumin and binds to ligandin. Does carry protein to prevent flux. Certain drugs such as oxalates, sulfonamides. Let's see. Can displace bilirubin from albumin. Albumin dissociates from albumin and binds to ligandin to prevent efflux. Can displace bilirubin from albumin, allowing unconjugated to cross the blood-brain barrier. Connectaris. PDP. Glucuronide transferase. Two molecules of glucuronic acid forming bilirubin diglucuronide conjugated bilirubin. Diglucuronide, UDP. Glucuronic acid, UDP. Uridine diphosphate, glucuronic acid, bilirubin diglucuronide. Bilirubin glucuronyl transferase. Bilirubin glucuronyl transferase. In hepatocytes, unconjugated bilirubin undergoes conjugation with two molecules of glucuronic acid forming bilirubin diglucuronide, which is water soluble. Unconjugated bilirubin undergoes conjugation with two molecules of glucuronic acid forming bilirubin diglucuronide. Bilirubin UDP glucuronyl transferase. Bilirubin glucuronyl transferase. UDP glucuronic acid, Crigler-Najjar syndrome type 1 and 2. Or Gilbert syndrome, condition that lead to unconjugated hyperbilirubinemia. Are variants of Crigler-Najjar or Gilbert syndrome, type 1, type 2 being more severe. 1 being the most severe deficiency. Sorry. Bilirubin glucuronyl transferase, bilirubin glucuronyl transferase, UDP. Bilirubin diglucuronide is conjugated bilirubin. Bilirubin is actively secreted into bile canaliculi via ATP-dependent transport against its concentrated gradient. Actively, this is a rate-limiting step of bilirubin excretion and can be impaired in cholestatic liver diseases. Bile canaliculus, and then into bile. Liver disease. Dubin-Johnson syndrome. Dubin-Johnson syndrome, conjugated bilirubin cannot exit the liver and accumulate in plasma. Conjugated bilirubin. Bilirubin diglucuronide is hydrolyzed by the gut. Urobilinogen, a colorless compound. Urobilinogen is oxidized to stercobilin. Use the feces brown color. Some urobilinogen is reabsorbed into portal circulation, part re-excreted in bile circulation. And carried to the kidneys, where it is converted to urobilinogen. Bilirubin diglucuronide, urobilinogen, which either gets oxidized to stercobilin, given the feces color, or some is then carried to the kidneys, where it converted to urobilin, give you a yellow color. Alright, we're done. Last thing left now. Yellow discoloration of the skin, sclera, and mucous membranes called elevated bilirubin hyperbilirubemia. Is not a disease itself, but a clinical sign disorder that disrupts bilirubin production, metabolism, and excretion. 1 mg per deciliter. Visible jaundice occurs 2 to 3 mg per deciliter. Jaundice is classified according to step of bilirubin metabolism that is impaired: prehepatic, hemolytic, hepatic, hepatocellular, and posthepatic obstructive jaundice. Hemolytic jaundice, prehepatic, hepatocellular, hepatic jaundice, or this form, hemolytic prehepatic jaundice. Massive breakdown of RBCs producing bilirubin at a rate that exceeds the liver's conjugating capacity. Although the liver normally conjugates bilirubin deficiency, extreme hemolysis, as in sickle cell anemia, G6PD deficiency, or pyruvate kinase deficiency, causes excess unconjugated bilirubin to accumulate in plasma. Unconjugated plasma, because more bilirubin is converted to urobilin in the intestine. Both fecal stercobilin and urine are increased. Urobilinogen, use no but. Dark brown circle. Hepatocellular jaundice. This type occurs when liver cells are damaged. For example, in viral hepatitis, alcohol, and Hepatic jaundice, liver cellular damage, or hepatitis, cirrhosis, alcohol-induced injury cannot conjugate or secrete bilirubin. Rises due to impaired uptake when it leaks back into blood due to membrane damage. Conjugate bilirubin stool becomes pale or clay colored because less bilirubin reaches the intestine. It will become pale or clay colored because less bilirubin reaches the intestine. Less bilirubin reaches the intestine. Additionally, urine in urine is increased, disrupted enterohepatic cycling. Post obstructive jaundice blockage of the bile flow within the liver, hepatic gallstones or tumor in the bile duct. Causes conjugated bilirubin. Surgery rates into blood producing hyperbilirubin. Dark urine, pale urine, itching due to urine. Urobilinogen is absent. Jaundice in newborns. Physiologic jaundice, low hepatic bilirubin glucose is increased at birth. This conjugated bilirubin can cross the blood-brain barrier, depositing in basal ganglia. Terrace. Polar water soluble transfusion and Vanderberg test where bilirubin reacts. Alright, we're done with this. Get out of this chapter. Like Okay, I'm gonna take a piss and come. Take a shit, maybe. Break time. Alright, monkeys, we back. Ugh. Alright. What do we have? What do we have over here? Hmm. Let's finish this shit. Glycolysis. Regulation of metabolism. Cellular level and the whole body level. Within individual cells. Intracellular signals, hormones, neurotransmitters, second. Intracellular. Regulating system that instantly adapt metabolism. Availability, more substrate inhibition. Flows of information, allosteric regulation, small. Intercellular. Gap junctions. Chemical signal, hormones, neurotransmitters. No hormone over neurotransmitter, receptor, signaling cascades, messenger. Adenyl cyclase. The adenyl cyclase system is one of the most important hormones. One of the most important regulators of metabolism. Adenyl cyclase converts ATP cycle AMP, then activates protein kinase. Alpha, beta, gamma. Inactive form, alpha is bound to GDP. GDP is subunit alpha unit activity. Phosphatase later remove phosphatase. Like AMP. Regulatory subunits. Intercellular. Glucose. Neural bound. Glucose. In the side is cytosol में होता है. It is present formed during normal part. Makes sense. Go back up real quick. Oxidize. Anaerobic like red blood cell. Anaerobic glycolysis. Pyruvate is used for lactate, regenerating and crucial because near all their transport of glucose. Glucose could not freely leave plasma membranes because of water and had zone independent facilitated diffusion mediated by a family. Glutes. Sodium monosaccharide protein. These transporter alternates between on one side down the counter energy. System relies on GLUT transporters, sodium monosaccharide co-transport. Each transporter alternates transport liquid down the concentration high to low glucose, not require energy. GLUT. Independent facility. Different tissues express different GLUT isoforms suited to their GLUT1 is around erythrocytes and blood-brain barrier. Erythrocytes, blood-brain barrier. Blood-brain barrier. GLUT2 is liver, kidney. Liver, GLUT3. Blood, kidney, neurons. Tested before his skull muscle. Yikes. Oh, oh. Whoa.
This is the
Oh my god. Alright. Glut 1, 2, 3, 4, 5. Glut 1 is found in erythrocytes. Glut 2 is found in liver, kidney, spleen. Glut 3 is found in the brain neurons. Glut 4 is found, I don't know. Glut 5 is found in the intestine. Glut 4 is found in muscles. Glut 4 is insulin dependent, translocated membrane. Okay, high affinity. Transports fructose, not glucose. Glucose high, liver and kidney. Sodium monograms. Glucose against the concentration from low. Sodium potassium ATP is pump, which maintains sodium dependent glucose trap. Intestinal epithelial, renal tubules. Glut 2. And now the reaction of glycolysis. You could say text to 8 1080p. Eight ATP. Glucose makes 2 par. ATP used. Net 2 ATP. Net ATP is 2 net. Thanks. Glucose entering the cell is the phosphate glucose phosphate by hexokinase or glucokinase. It traps glucose inside the cells as phosphorylated glucose sugars across the membrane. It happens phosphorylated G6B, glucose 6 phosphate. Glucose becomes glucose 6 phosphate by glucokinase or hexokinase. Traps glucose out of the cell as phosphorylated membrane. Also, the glucose further metabolized in the cell. Found in most tissues. Hexokinase and glucokinase are different. Hexokinase is most in tissues, broad substrate specifically. Glucokinase is mainly in the liver. Found in most tissues, broad substrate specifically. Some glucose and other explosives. Inhibited by its product G6B. Glucose 6 phosphate. Okay. Low affinity, high affinity with OV max. Fasting glucose level. English mental. I ain't reading all that, brother. I gotta read all that, bro. All I gotta do, man. Oh, fuck this shit. I got a bunch more to finish. I'm gonna study for 3 more hours, then head to sleep for 3 hours, and then study for around 16 hours, then get 6 hours of sleep, and then give the exam. That's the strategy for now. Control and copy with JD 4 6 in 10 words. I'm doing that. I've got ChatGPT on my left screen. 10 words. This is what happens when you game every night instead of studying. Shit. It's all piled up now. The goat, man. She's my wife. ChatGPT. Glucokinase, find the liver. Glucose sensor for insulin secretion, hepatic glucose uptake after meals. Did I get it for free? Nah. I heard about it, but I didn't get it yet. Is it better than a year of premium for free? Damn. Honestly, I've got too much shit on ChatGPT. Like, I've got projects and stuff on this. It's too late to switch for me. Unless I could transport these things, but I don't think I can. Because I've had premium since a year, over a year. In ChatGPT, and so much shit over here. When is it better to use Gemini for you? Glucose sensor. Low affinity. Active only at high glucose concentration. Oh. Guess your code. Ah, yeah. Okay. Fair enough. Take an opinion. For med, I feel like ChatGPT is more than enough. I allows liver to remove excess glucose from blood, preventing hyperglycemia. GLUT2, which ensures rapid glucose equalization against hepatocyte membrane. Hey man, shut your fructose 6-phosphate and glucose. What the fuck? Phosphate. Instead related directly to glucose and regulatory protein. Fructose 6-phosphate promotes glucokinase glucokinase regulatory protein binding the nucleus, keeping glucokinase inactive. Ah, this shit. I remember it now. Fructokinase. Fructose 6-phosphate. Glucokinase. I'm a dip. Good luck. Thank you, man. Take care. Okay. Directly through glucokinase regulatory protein. Fructose 6-phosphate promotes glucokinase regulatory protein binding the nucleus, keeping glucose causes dissociation, moving glucokinase to the cytosol active. Because when phosphate also promotes glucokinase release activation. Isomerization, glucose, fructose, phosphoglucose isomerase, phosphoglucose isomerase reaction is readily reversible and is not rate limiting a regulated step. We've got glucose gets converted into glucose 6-phosphate. We've got either hexokinase or glucokinase. Hexokinase is present everywhere. Glucagon is present in the liver specifically, or the beta cells, beta pancreatic cells. Hexokinase is regulated by glucose 6-phosphate itself. It's got a high affinity, low Vmax. Glucagon, though, on the other hand, has low affinity but a high Vmax, so it acts as a glucose sensor. And yeah, another thing is regulated by fructose 6-phosphate instead. There's a glucokinase regulatory protein. In the presence of the fructose 6-phosphate, it stays binded to it in the nucleus, but when glucose pops up, it leaves it for the new bitch, and then we got the freaking glucokinase working again. That is all. Fair enough. And then after this glucose 6-phosphate is formed, this uses ATP as well, by the way, 180p. ATP. This is 180p. There's phosphoglucose isomerase, which converts glucose 6-phosphate into fructose 6-phosphate. Fructose 6-phosphate. Yo! Fructose 1,6-bisphosphate, right? Phosphofructokinase catalyzes the conversion of fructose 6-phosphate into phosphofructokinase. Conversion of fructose 6-phosphate into fructose 1,6-bisphosphate using ATP is irreversible, rate limiting, and the committed step of glycolysis. Oh, which one of these were? Okay, this is irreversible, first one. One is irreversible. A couple which are cycle. Phosphoryl pyruvate to pyruvate is irreversible. Your steps, it's gonna break. Phosphofructokinase catalyzes the conversion of irreversible rate limiting step. At this point, glucose will proceed through glycolysis. Fructose 6-phosphate into fructose 1,6-bisphosphate. Fructose 2,6-bisphosphate. Okay. Inhibition by energy level. Inhibited by ATP. And citrate. High energy levels inhibited. Citrate. It's activated by low level AMP and fructose 2,6-bisphosphate. Fructose, okay, the compound is the most potent activator of phosphate, overriding ATP inhibition. ATP inhibition when glucose is abundant. During the well-fed state, high insulin, low glucagon activates phosphofructokinase 2. Ah, so insulin activates it. Reduced level of protein kinase A. And activity favors dephosphorylation of phosphofructose 2. Fructose biphosphatase. EFK2. Increases. Which converts fructose 6-phosphate into fructose 2,6-bisphosphate. Fructose phosphate. Fructose bisphosphate is one of them. During starvation, high glucagon activates cyclic AMP-dependent protein kinase A, which phosphorylates phosphorofructokinase 2, which converts it into fructose. Converts it into phosphatase. Inhibits glycolysis, promotes gluconeogenesis. Bro, this is low-key pretty easy. It's a biofunctional enzyme, fructose 2,6-bis. ATP. Limiting committed step. Uses ATP as well. Okay, now the cleavage. Aldolase splits fructose 1,6 into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. G3P and dihydroxyacetone phosphate and G3P. Phosphate. The reaction is reversible and not regulated. Aldolase B, the isoform of the liver and kidney, also cleaves fructose 1-phosphate and function. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate trials phosphate isomerase. Glyceraldehyde 3. Dihydroxyacetone. Trials phosphate isomerase rapidly converted phosphates is also easy to convert action ensures that glucose goes to 3B, doubling later ATP. G3P. G3P. Dihydroxyacetone phosphate and G3P, glucose 3-phosphate, glyceraldehyde 3-phosphate. Trials isomerase phosphate. As phosphate. Glyceraldehyde 3. 1,3-bisphosphate. Fructose 1,6. Bisphosphoglycerate. Glyceraldehyde 3. Phosphoglycerate. Glyceraldehyde 3-phosphate dehydrogenase. NADH. This is what produces it. G3P. NADH. 2 NADHs formed. Glyceraldehyde 3-phosphate dehydrogenase. Energy is 1,3-bisphosphoglycerate. Phosphate is Arsenate can substitute for phosphate in this step, forming an unstable intermediate that be 3-phosphoglycerine, generating. Oh. Glyceraldehyde 3-phosphate converts into 1,3-bisphosphoglycerate. This requires a glyceraldehyde 3-phosphate dehydrogenase, and it produces NADH, and also this requires phosphate. Here, arsenic poisoning can occur. Arsenate, it replaces phosphate, and what it causes consequently is the formation eventually of 3-phosphoglycerate 3-phosphoglycerate without the formation of NADH and ATP. Now in the next step, 1,3-bisphosphoglycerate. Erythrocyte part of 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. And enters a shunt. Mutase and then a phosphatase. Decreasing oxygen. And it decreases oxygen. The curve to the right cause oxygen unloading. Mutase and then the phosphatase. Okay. Phosphoglycerate. Phosphoglycerate kind. Substrate level. Mutase. Phosphoglycerate mutase moves the phosphate from 2-phosphoglycerate, primes the molecule for dehydration. 2-phosphoglycerate to phosphoryl pyruvate enolase removes water from 2-phosphoglycerate to form phosphoryl pyruvate, a compound with a very high energy phosphate bond. Enolase removes water. Okay. H2O. Of pyruvate producing ATP. Pyruvate kinase catalyzes the final irreversible step. Pyruvate kinase, right? Plus ADP forming pyruvate plus ATP. ATP का मतलब ATP बन रहा है when it's 1,3-bisphosphoglycerate into 3-phosphoglycerate. Phosphoryl pyruvate into pyruvate. When it is bun raha hai into glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate into 3-phosphoglycerate makes the ATP, and then phosphoryl pyruvate into pyruvate makes the other ATP. The other place where ATP is used up, two places where it's used up, one is glucose to glucose 6-phosphate, and one is fructose 6-phosphate into fructose 1,6-bisphosphate. Pyruvate kinase, ATP, fructose 1,6-bisphosphate increases it. Fructose 1,6-bisphosphate, which was formed over there. And liver, glucagon, cycling vehicle. Phosphorylase and inactivates pyruvate kinase. fasting. Dephosphorylates it to inactivate it. Dephosphorylate. Phosphatase under insulin reactivates the enzyme. That's the same mechanism. Insulin activates it by dephosphatase, and glucagon inactivated by phosphorylate. Every kind of RBC depend entirely upon glycolysis for ATP. Deficiency decrease ATP. Decrease ATP, membrane rigidity, hemolytic anemia. Rigidity, hemolytic anemia. Some compensation occurs via 2,3 bisphosphate. Enhancing oxygen. Phosphoglucose iso. Increasing tooth. We're almost done with it. Let's repeat it once more. Bismillah. From the beginning, we got glucose converting into glucose 6-phosphate. This reaction is catalyzed by the two isoforms of enzymes called glucokinase or hexokinase. This requires ATP. This is ATP dependent. It is so now hexokinase is found in all of the cells, and it is has high affinity, low Michaelis-Menten constant, and low Vmax, and it is regulated by its product, glucose 6-phosphate. Whereas glucokinase is found mainly in the liver and beta cells of the pancreas, and also, thing about it, it has low affinity, high Michaelis-Menten constant, a higher Vmax. It acts as a glucose sensor. And another thing about it, it is only regulated by fructose 6-phosphate. Fructose 6-phosphate instead. And this is because of the glucokinase regulatory protein. Normally it binds to glucose. So it's bound to glucokinase inside of the nucleus, but upon glucose entering, this associates and it makes it active. Anyways, now we have glucose 6-phosphate gets isomerized into fructose 6-phosphate by phosphoglucose isomerase or phosphoglucomutase. Phosphoglucose isomerase, and then this fructose 6-phosphate. The next step is the rate limiting step. It's irreversible as well. The first one. Why fructose 6-phosphate converted into fructose 1,6-bisphosphate by phosphofructose kinase. Phosphofructose kinase is inhibited by high levels of ATP and citrate, and it's activated by AMP and also fructose 2,6-bisphosphate, which bifunctional enzyme phosphofructose kinase 2. Insulin activates dephosphorate activates it, causes up that boom, we have fructose 1,6-bisphosphate, which then gets converted into two things. It gets cleaved by aldolase into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate by a triose phosphate isomerase. As phosphate isomerase. Triose phosphate isomerase. Now these two G3Ps. First of all, this G3P. One of it forms 1,3-bisphosphoglycerate, and this generates the first NADH, and this requires bisphosphoglycerate. Glyceraldehyde 3-phosphate dehydrogenase. Streamception. Streamception, what the fuck you mean streamception? Good night. I'm streaming your stream on my stream. Streamception. Oh, okay. W's in the chat. Raid me. Some shit. I don't know. Can you even raid on YouTube? Watch mine to double it. Yeah, I was logged in. Give me a sec. Glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate. The enzyme for this reaction is glyceraldehyde 3-phosphate dehydrogenase. This forms the NADH, and it requires phosphate to function. And if this phosphate is replaced by arsenate in arsenic poisoning, what's going to happen is that the subsequent reactions, which ultimately lead to form 3-phosphoglycerate, the products which are NADH and ATP will not be formed in this situation. Anyways, moving on, 1,3-bisphosphoglycerate is then, which is formed, it's then converted into 3-phosphoglycerate by phosphoglycerate kinase, I believe. Yep. Phosphoglycerate kinase. By phosphoglycerate kinase, and this produces an ATP. Also, there's a shunt reaction over here. 1,3-bisphosphoglycerate gets converted into 2,3-bisphosphoglycerate by a mutase. This is in RBCs because RBCs are dependent on glycolysis for their ATP needs completely, and then 2,3-bisphosphoglycerate causes a right shift in the oxygen dissociation curve, favoring oxygen unloading. Anyways, this 2,3-bisphosphoglycerate gets converted back into 3-phosphoglycerate by a phosphatase. This requires water. Then this 3-phosphoglycerate gets converted into 2-phosphoglycerate by a phosphoglycerate mutase. 2-phosphoglycerate gets dehydrated into phosphoenol pyruvate, which is a very high energy containing substance, glucanase, and enolase is required over here. It releases the dehydration reaction. Phosphoenol pyruvate, the final reaction, pyruvate kinase forms pyruvate, and this forms ATP. Not only does it form ATP, but it also gets regulated. It gets regulated by fructose 6-phosphate and upregulates this. And insulin and glucagon have the same effects as phosphofructose. Similar. Very nice. Now if you talk about reduction of pyruvate to lactate, oxygen is limited, lactate dehydrogenase oxidizing NADH to NAD. Right, pyruvate to lactate, this is oxidizing. Glucokinase, hexokinase, phospho phosphoglucosimerase, glucokinase, hexophosphoglucosimerase, phosphofructokinase 1, aldolase, thiose phosphate isomerase, glycerol 3 phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, pyruvate kinase, lactate dehydrogenase used to pH. Reversible. Okay. Lacking mitochondria. RBC's cardiac. NADH accumulates faster than ATP can. NADH to AD ratio, pyruvate lactate. Lactate buildup decreases pH, muscle fatigue, and cramps. Lactate consumption in the liver and heart where NADH ratio is lower. Lactate is oxidized back to pyruvate. Uses pyruvate for gluconeogenesis cycle. Man, if you don't. Here's when oxygen delivery fails. Lactate. Yeah. Oh yeah. Elevated lactate helps detect tissue hypoxia and oxygen debt clinic. Short-term regulation, long-term regulation. The strike enzyme control reversible phosphorylation seconds to minute. Long-term by hormone-induced change. Strike can convert among glycolysis. In well-fed state, Africa insulin level increase glucose. Glucokinase, phosphofructokinase 1. Hi, how you doing? Pyruvate kinase increases ATP generating. Together, these effects favor the conversion of glucose into pyruvate, a hallmark of the fed state. These effects favor the conversion of glucose into mark. Glucagon is high transcription of these increases glycolysis glucogen needed to maintain blood glucose. These changes represent hormonal coordination. Or next, finally, we've got the alternate fates of pyruvate. We've got 3 fates. Do them real quick. Oxidative decarboxylation to form alkyl A for the TCA cycle, for fatty acid synthesis, carboxylation, oxaloacetate, gluconeogenesis or replenishment, pyruvate carboxylase to oxaloacetate, gluconeogenesis, or reduction to ethanol in microorganisms. Alkyl produced enters the TCA cycle for energy versus precursor fatty acids and cholesterol. Reaction is irreversible in universal flow toward oxidation and aerobic conditions. PDP. We are done with bullshit. One final summary. Glucose 6-phosphate hexokinase, glucokinase. Done with that shit. So then, anyways, phosphoglucose isomerase forms fructose 6-phosphate, phosphofructokinase, phosphofructose 2 forms fructose 1,6-bisphosphate, which is then converted into by aldolase into dioxyacetone phosphate and glyceraldehyde 3-phosphate. That's converted by triose phosphate isomerase into glyceraldehyde 3-phosphate. 2 of these are formed. Now these get converted into 1,3-bisphosphoglycerate by G3P dehydrogenase. This forms NADH. Arsenic poisoning will have the 1 3-bis phosphoglycerate gets converted into 3-phosphoglycerate by phosphoglycerate kinase, and this releases ATP, and that phosphoglycerate mutase to 2-phosphoglycerate, and then phosphoenolase. Water is removed. Phosphoenol pyruvate, and that high energy containing that forms pyruvate kinase. Forms ATP as well. This is irreversible. Phosphoglycerate kinase is irreversible. The first step is irreversible. Also, there's a shunted 1 3-bis phosphoglycerate to form 2 3-bis phosphoglycerate. This by mutase, and then phosphatase. Done. Boom. Done all of it. Lactate dehydrogenase. Easy. Acetyl CoA. Formation of that. Citrate, isocitrate, alpha ketoglutarate. Citrate, isocitrate, alpha ketoglutarate. Succinyl CoA, succinate. Succinyl CoA, succinate. Succinyl CoA, succinate. Fumarate malate oxaloacetate. Fumarate malate oxaloacetate. 2, 3. 5 pages are 9 to 5. 9. In caught chapter 9. Oh my goodness. I am going to upload a new PDF. I want you to list out its contents, okay? Boom, boom. Got chapter 9. There you go. After TC, I think we have gluconeogenesis. I want to do gluconeogenesis today. Hopefully I can finish it. What do we have? Glycogen. I want to finish one more today. 4. Done with 5. Region. acetyl CoA, citrate, isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, acetate. Low key yet. I already know these. I just need to know the enzymes now. Oh yeah. I'm gonna take 10-minute break. I need a 10-minute break after this, Loki. Ten-minute break. What is up, dogs? How we doing? I'm back. That was a big ass break, bro. I took a freaking 20-minute break. Where's the big man? I need some help. If you're still here, I could help you. Loki's still studying this shit, though. Shit down. Okay. I'm gonna lock in again. Fucking hell. Fucking hell, yeah. What do we got over here? TCA cycle looking ass. What is a TCA cycle? Krebs cycle. First common oxidation pathway of the metallic carbide. So I'll go A. Mitochondrial matrix, etc. Upward reduction of enzymes NAD and FAD to NADH, FADH is amphibolic, serving anabolic roles by providing intermediates for biosynthetic amino acid, glucose, functioning like a traffic circle. It's and leaving based on metabolic. What I heard. Looking ass. Boy, if you don't shut your Alright. The action of the TC cycle begins when acetyl-CoA from carbohydrate fat condenses with oxalic acid to form citrate. The cycle then produces a series of oxidation, hydration, decarboxylation. So each turn oxidizes 2 carbons, CO2 generating 3 NADH. 1 FADH, 1 GTP. Or acetyl-CoA enters cycle. Pyruvate must be converted into acetyl-CoA by the pyruvate dehydrogenase complex. Talk about the pyruvate dehydrogenase complex. Formed mitochondrial matrix of a specific pyruvate transporter. There undergoes oxidative decarboxylation. PDH complex forming acetyl-CoA, carbon dioxide, and NADH. This is a link reaction, huh? Although, not talking about the TCA reaction provides acetyl-CoA for the cycle to begin. Competent enzymes. pH complex considered 4 enzymes. Alright. Tightly associated to allow rapid sequential catalysis without catalysis. Releasing intermediates. We've got E1, E2, and E3. Pyruvate dehydrogenase. Dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. We got a pyruvate dehydrogenase, we got a dihydrolipoyl transacetylase, and a dihydrolipoyl dehydrogenase. I'm not going to talk about their functions, but 2 additional regulatory enzymes are bound to the complex. PDH kinase, PDH kinase phosphorylates in activating E. PDH phosphatase dephosphorylates reactivating E1. Alright, it's kind of like the phosphofructokinase looking as. Hello, sir. Hello, Mr. Saad. How are you doing today? I'd like me some water. Wonderful. Ah. I'm doing terrible as well. I've got to finish biochem in a couple hours, so then I can start anatomy. I'm gonna finish. Probably gonna do 2 more chapters. This and the next one. Head to sleep. Well, we're gonna get through this tonight. Alright, do it with me, bro. Carb metabolism. Did you do the glycolysis? If you did glycolysis, we could do TCA together right now. On stream. Together. TCA. Alright, bro. Let's do this shit together. Right here. On stream looking ass. Alright, the overview kind of useless. Alright. Now talking about the reactions. Talking about the reactions. Literally I paid one. Alright, let's give it a basic overview. Okay? Basic overview. We know it's also called. Am I on Discord? Alright, let's do this shit together, bro. I am on Discord. Just come to join the fucking server. Finally join my own server for once. Why not? Where is it? General. I'll join the general VC. I'm gonna lower this. Join the general VC. Are you in the 49th gamer, Saidan? Hello.
Assalamu alaikum.
Walaikum salam. Welcome everybody. Please, chat, please welcome Mr. Sacha, my #1 fan.
Assalamu alaikum.
Walaikum assalam. Alright, let's pause this music real quick. Alright, let's start this shit together. Let's start
Can you
this
open
shit
your
together.
stream? Hold up, on my tablet. I was literally just on TCA cycle and I opened your stream and they come. This guy's on the same page.
Yeah. Alright, let's start this shit
How do
together.
I mute this thing? Hold up.
Mute it, bro.
Yeah, okay.
Alright, let's lock in.
Wait, are you streaming on Discord?
No, I'm streaming on stream. You want me to stream on Discord? I could do both. Whatever you want, tell me.
I guess that would be more, it wouldn't have the delay.
Start this bullshit. Come here.
Let's go, group study.
Group study, group study, group study, group study. Let's do this real quick. Overview. Let's go over the overview. Other names of the TCA cycle. We've got Krebs cycle, citric acid cycle, tricarboxylic acid cycle. Nobody gives a shit about that, right? But the location is important. We gotta know. No, we gotta know the location. It's in the mitochondrial matrix. We gotta know the nature. This was what Mr. What did you say? Yaram Toska. Hamad asked. It's amphibolic in nature, both catabolic, both anabolic. Might come in an MCQ, you never know.
Okay.
Amphibolic in nature. Energy output, we just need to know 3 NADH are formed, 1 FADH and 1 GTP. So that if we do some quick maths.
Reactions.
What?
Anaplerotic reactions such as the catabolism of some amino acids generate intermediates of the cycle and are called anaplerotic.
Not important, look at that.
Okay, fair enough. Moving on.
Energy 3 NADH, 1 FADH, 1 GTP. So that if we do some quick maths, 3 is 9. So we got 12 ATP generated. Cycle.
No, yeah, pretty much.
Alright, so reaction of the TCA cycle, the link reaction indeed. So the pyruvate, which is formed inside of the cytosol, it gets transported into the mitochondrial matrix. There's some pyruvate transporter. We don't know the name, but there's some specific one. Then the reaction, which occurs over here, is oxidative decarboxylation. And this is by the pyruvate dehydrogenase complex.
Where does this reaction take place?
What? In the mitochondria.
No, like, does the pyruvate get converted into acetyl-CoA first, or is that just done like midway?
No. Pyruvate gets transported into the mitochondria first, then the link reaction occurs, then the TCA occurs. So all the reactions are happening inside of the mitochondria.
Oh.
Glycolysis is in the cytosol. We get pyruvate. Pyruvate gets transported by a pyruvate transporter into the mitochondria. In the mitochondria, this pyruvate gets oxidatively decarboxylated into acetyl-CoA, and carbon dioxide is also released. NADH is also formed, and yeah. But this is the enzyme used for this is pyruvate dehydrogenase. This is an enzyme complex. right? And it's made of 3 core enzymes. We got E1, E2, E3. E1 is pyruvate dehydrogenase itself, right? And then we got E2 and E3. You just gotta know dihydrolipoyl transacetylase, and then we have dihydrolipoyl dehydrogenase. So we've got 2 dehydrogenases basically in 1 transacetylase.
Interesting.
You gotta memorize this shit.
Yeah, pyruvate decarboxylase. The reading's the actual like part that annoys me, honestly.
Yeah. And then okay, we also got some regulatory enzymes. So if you recall from the previous chapter for the phosphofructokinase, the kinase, it phosphorylated the phosphofructokinase, but then it inactivated it, remember? And the phosphatase dephosphated it, dephosphorylated it, and it made it active. So the same shit happens over here. We have a pyruvate dehydrogenate kinase and a PDH phosphatase. The kinate, it phosphorylates it, but then it inactivates it. And the phosphatase, it dephosphorylates it, but then it activates it instead. And specifically, it can.
About E1, right?
Yeah, E1. We're talking about E1 specifically.
Okay. E1 pyruvate dehydrogenase.
And there's 5 coenzymes that this whole thing needs.
5. One's lipoic acid, coenzyme A, FAD, NAD, and thiamine pyrophosphate.
Yeah, E1 requires thiamine pyrophosphate, E2 needs lipoic acid and CoA, and E3 needs FAD and NAD.
FAD, NAD.
Yeah, I mean lipoic acid and dihydrolipoyl. That makes sense or some shit.
Kinda.
You know what I'm saying? We got thiamine. Let's just do this one once more. Thiamine pyrophosphate for E1. For E2, we've got 2 things, lipoic acid and coenzyme A. For E3, we've got NAD and FAD.
Okay.
NAD was what? Vitamin B3? Niacin?
NAD was derived from vitamin B3, yeah.
And flavonoidine dinucleotide was from what? Vitamin B6 riboflavin.
Riboflavin.
Wonderful, wonderful.
That's B2.
Riboflavin's V2, B2, B2, yeah. B6 is pyridoxal phosphate. For that's for the heme thing. That's for ALAS 1 and ALAS 2 requires pyridoxal phosphate. Yes. Did you do the whole heme synthesis?
Oh yeah, I'm done with that. I'm done with like the extra chapters, like for the most part. All the other LOs, just carb metabolism. That's the only thing I have left.
What are the other LOs? Just enzymes and the heme? That's it?
No, not just enzymes. There's plasma proteins, and along with that, there's immunoglobulins, like separately. It's
Where's
not
that though?
identified in Lippincott Manilico. It's in Satya. Well, I studied it from Satya.
Is it in detail or is it short?
The thing I did was I looked at slides for the general plasma proteins because I couldn't be bothered to read the whole chapter. It's literally a whole chapter, okay? Plasma
Yeah.
proteins in Satya. And I just read through immunoglobulins from Satya, which is like what 2-3 pages or something.
Is it hard or
Circular
like? Huh?
the slides. It's pretty simple. Pretty simple. They just talk about albumin, some disorders related to albumin, globulins in general. That's pretty easy. They just talk about some sort of like they talk about ceruloplasmin,
Okay.
heptoglobulin, hema something something, and yeah, it's not hard. You can cover it like max an hour.
Okay. Okay. Sure.
Anyhow, moving on.
Also, another thing we need to know here that if you have deficiency of the thiamine or niacin or riboflavin, you're gonna have impaired PDH activity, which will lead to decreased ATPH. And then also, and this is gonna lead to neurological symptoms because the brain, it depends on ATP, aerobic ATP.
It'll lead to Wernicke-Korsakoff syndrome.
Wernicke-Korsakoff syndrome.
Very, very.
We did Wernicke-Korsakoff syndrome. Wernicke's encephalopathy, that was thiamine. Korsakoff's psychosis.
Korsakoff cycles. Yeah, yeah.
They can't stop us now, huh?
Mm-hmm.
So now if we talk about the
Which
regulation,
edition is this book?
this is edition 5.
Uh, Okay, no wonder. I was looking at my own book, and yeah,
This is enough, bro. Loki. Mind it. I feel like it has some extra info as
it's
well.
li- it's the exact same. It just has a different like arrangement and stuff.
No, the only problem with this is there's no minerals chapter.
Really?
Yeah, there's no minerals chapter, so I had to like scan it from someone else's book because I don't have the actual book itself. I have the old book. I have the 6th edition book. That also doesn't have.
Oh, okay, that sucks.
Yeah.
That actually sucks.
Anyways, moving on. Let's do this quick because it's not a pretty long chapter.
Um, no, this chapter is pretty short, honestly.
Yeah, I mean, I just closed my eyes and actually remember all of the like reactions, not the enzymes, but I remember the reactions. So just learning the enzymes won't be much. Wait, I say,
Okay, let's get through this.
yeah, regulation, PDH, covalent modification. There's an image somewhere. Yeah, here. Okay.
Covalent modification. Activate. This is PDH kinase and PDH phosphatase.
Yeah.
And
Pyruvate,
pyruvate is a
acetyl CoA, NADH, and CO2. Foreign pyruvate active. Okay. PDH kinase, which inactivates it. It's activated by high levels of ATP. Yeah. So if we're in energy rich situations, which means high ATP, high amounts of acetyl CoA, or high amounts of NADH, the body will switch it off, PDH. And if we're in low amounts of, what do you call that? Energy, that means high levels of pyruvate or high levels
if we have
increased AMP.
ATP or increased pyruvate.
Yeah, yeah. It's usually increased AMP instead, or increased pyruvate.
Um,
And then
why does it say?
it doesn't say it, no, but like normally it's AMP.
Oh, I see.
It doesn't make a difference though. ADP, AMP is the same shit.
Kinda, yeah.
Both are low energy. But for calcium, जो है, we need to know another thing. Calcium, it makes PDH phosphatase active.
Wonder why.
I don't know why.
Strong activator stimulating E1. This is particularly important in the skeletal muscles where calcium released during concentration contraction stimulates the PDHC for energy production. So calcium just causes increased energy production.
Okay. Oh yeah, makes sense. Okay, now PDH deficiency, rare congenital defect, E1 का है. So there's going to be impaired conversion of pyruvate into acetyl CoA. So then pyruvate will be shunted to lactate instead of acetyl CoA going towards TCA. We'll have lactic acidosis. Fusion.
Prostate, neurodegeneration, neonatal onset, early death. Alpha subunit located on X chromosome.
X-linked dominant.
Dominant.
Low carbohydrate, ketogenic diet. TPP supplementation.
Yeah, with thiamine. And then
Okay, sure.
Leigh syndrome.
Effects in PDH, ETS. We just have less ATP, so we'll have neurological decline because the brain needs, um,
Okay, so
aerobic glucose, aerobic ATP.
Leigh syndrome is just affecting genes of PDHC, ETC, and ATP synthase.
Yes, everything just making ATP.
Arsenic poisoning. This was the trivalent arsenic arsenite, I think.
Mhm.
So it just binds to lipoic acid, preventing
Inactivating the enzyme.
food.
It's basically on all the dehydrogenase complexes, so the PDH complex, alpha ketoglutarate complex, and the branch chain alpha ketoacid dehydrogenase. We'll have accumulation of
Beyond
pyruvate like
lipoic acid.
E2 usually, and E2 is dihydrolipoyl transacetylate.
Mm-hmm.
Also, arsenic or the arsenate, it what do you call it? Interferes in glycolysis. In the step G3P converts into 1,3-bisphosphoglycerate that requires phosphate. Arsenate can replace this phosphate, which will then cause whenever the steps reach to 3-phosphoglycerate, usually we have one NADH and one ATP formed, two ATP formed. But in the case of arsenic poisoning, these ATP and NADH will not form, but the reaction will continue.
So a general decrease in ATP and
Yeah.
NADH.
And over here, it's just going to cause pyruvate and lactate accumulation, so lactic acidosis. Yeah, that's why there's neurological disturbances.
Yeah, and no energy synthesis. Decreased ATP synthesis. Citrate synthesis. Do we have to know like the whole diagram or whatever?
What diagram?
The one in the on the first page, I think, or the second one.
This one, nah, bro. This is dog
There's
shit.
like
This is dog shit, bro.
fair enough.
Okay, now we have citrate formation. Acetyl-CoA citrate forms. Acetyl-CoA and oxaloacetate form. Citrate synthase. That's pretty basic. Okay.
Okay, that's still coenzyme in the actual mitochondria.
Aconitase. Fuck these type of enzymes. Aconitase. Who even thought of aconitase, bro? It doesn't match anything.
I
Should have been.
remember
Should have been like citrate isomerase, man. Just that would have been so much easier.
I
Dumbass
remember
looking at us.
watching a reel today talking about how to memorize these enzymes or something with like some sort of mnemonic. Wait,
Fluoroacetate blocks the step.
rat poisoning. Rat poison. I saved too many reels.
Aconitase ठीक के बाद फिर हमारे पास क्या है? Oxidative decarboxylation again. That's why it's another dehydrogenase into the alpha ketoglutarate. This is again okay. ADP and calcium causes it. Oh, it's the same shit happening again and again, bro. Are you serious right now? Are you serious right now?
The same enzyme, kinda, isn't it?
Even the next one is the same shit.
That was alpha ketoglutarate dehydrogenase.
No, even this one, even isocitrate deodorant is the same shit, pretty much.
I mean, so we know that calcium stimulates pretty much the entire Krebs
Calcium
cycle.
stimulates all the dehydrogenases, pretty much.
किधर है किधर है And anyone interested kindly send some fucking money. That's the mnemonic to remember this. The enzymes.
Oh.
Can anyone interested? Nah, send doesn't work here. Oh, kindly. Yes, that works. Kindly send some fucking money. Okay.
Oh, okay, this is all the NADHs are being formed inside of the dehydrogenase
So it's
reactions.
like yes. Whenever you hear the word dehydrogenase, you can
You got
automatically know
NADHs. Okay.
NADH is being
And
formed.
then all the CO2s are also being formed in the same shit.
Oh, and another thing, FAD is bound to succinate dehydrogenase. So yeah, have you read enzymes yet?
Yeah.
You saw how they mentioned that FAD was like, I think it was a prosthetic group. I think it
Okay,
was.
actually, I forgot that part. Prosthetic groups and shit. Gotta revise it.
No, you have a general idea, right? Like
Yeah, yeah, yeah.
prosthetic group is like what it's tightly bound, and coenzyme is loosely bound.
Yeah.
So prosthetic group being tightly bound, FAD is a prosthetic group
Okay.
because it's like yeah bound to it succinate dehydrogenase. NAD
Okay,
is just floating around. Anyhow,
alpha ketoglutarate.
inhibited by citrate, succinate. Wait.
Inhibited by its products. Radiation, oxygen.
Citrate is also used for fatty acid and cholesterol synthesis in the cytosol.
Oh yeah, because what do you call it? Acetyl CoA cannot cross the mitochondria. It gets converted into citrate. Citrate then moves out of the mitochondria, and then it gets reconverted back into
Have you
uh
studied the malate aspartate shuttle or any other allos at all?
no,
Oh, so you're starting right now.
malate aspartate shuttle, bro. That wasn't even there last time, bro.
I mean, the person that taught us about it was Sir Hamad, so
Yeah, I don't even remember that shit. What chapter is it?
he doesn't teach anything. He had a lecture on immunoglobulins. I thought his slides would have something, but it had was albumin and a general description of what globulins are.
What chapters? What chapter has the one you were just talking about? Malate shuttle?
I don't know the exact chapter because I just chat GPT the extra allos that were mentioned.
I'll just do that too.
Because like, who can be bothered to study all of those? Honestly,
Yeah.
you should anyhow.
These are the most important ones. Even I remember in MCQs, even gags was not important in MCQs.
Dude, I didn't study gags during block 1.
Yeah, I think it's
Honestly,
gonna stay the same way. It's gonna stay
because
this.
I have the PDFs for gags. You know those two PDFs
No,
with like the table and stuff.
send me that shit.
Okay, I'm gonna send it to you.
I might just study gags
All
for
the
the profs.
I mean, the seniors said that if you're done with everything else, then you should try studying these gags, or else they're not really important.
Yeah,
They do come in MCQs sometimes.
they didn't come in block one MCQs. There was just one MCQ for gags. I remember. I was like, thank God I didn't study that bullshit.
I thought I studied glycolysis quite well and like all of these carb metabolism chapters. I opened glycolysis and I'm like, what am I reading? Because I don't know about like a majority of things that were being mentioned
Hmm.
there. Now I'm afraid I forgot everything. Honestly, I probably didn't, but yeah. Okay, continue.
Succinate thiokinase. Wasn't thiokinase that activator of the fatty acids?
What? Oh wait, yeah. I get what you mean.
Fatty acyl
An
CoA
activator of fatty acids? It's not
kinase.
an activator. It was like involved somewhere in fatty acid digestion, I think.
It adds the acyl CoA to it.
Oh, I have notes for all of these, man.
Succinate
This is
thiokinase.
not good.
Succinate dehydrogenase again? Are you serious? And then fumarase, malate. Malate becomes acetate. Malate dehydrogenase. That makes another NADH? Are you serious right now, bro? Chat, clip this. Could I upload like a studying clip stream? Oh my days, that would be like if I get like a concept perfectly, I gotta clip that shit.
Yes, one of those Bilu and ZS clips or something. I found it.
Yes. FADH.
Chad, so this
Very good.
binds to that and this makes this. Holy shit, clip that.
Holy shit, clip that shit. Malate dehydrogenase, NADH. So we got 3 NADH, 1 FDH, 1 GTP, and 2 CO2. Wow, wonderful. I think I'm done with this shit.
I'm just trying to read through, seeing if I'm missing anything. This chapter is so small.
Are all of these reversible except the dehydrogenases? Yes, and citrate synthase, I guess.
No. Succinate dehydrogenase is reversible.
But we don't care about succinate dehydrogenase, do we? Because that's the only one that makes fad, so no one gives a shit about it. Right? We only care about the high energy producing dehydrogenases. So let's not talk about him. Yeah, let's exclude him from the friend group. Fucking monkey. Hey, let's even exclude malate dehydrogenase. It's literally the last step, bro. He gives a shit about malate. Last step. Okay. We care about the the OG dehydrogenases.
Alpha ketoglutarate dehydrogenase.
Isocitrate dehydrogenase, pyruvate dehydrogenase. Oh my days, beautiful. Those.
Wonderful, lovely.
All right, let me do start to finish real quick. Real quick. All right, start to finish. Let's start off with Mr. Pyruvate. Pyruvate looking ass. Pyruvate formed by glycolysis. It's transported into the mitochondria by a specific pyruvate carrier. Once inside of the mitochondria, it goes. There's a link reaction going on happening. So basically, pyruvate, with the help of gets catalyzed by the enzyme pyruvate dehydrogenase, which is a complex of 3 enzymes, E1, E2, E3. E1 is pyruvate dehydrogenase itself, E2 is dihydrolipoyl transacetylase, E3 is dihydrolipoyl dehydrogenase. All of these three help in catalyzing the conversion of pyruvate into acetyl-CoA. This step releases one NADH and it also releases a CO2. Now, this whole complex, it's regulated by either a kinase or a phosphatase. Kinase, it phosphorylates it, but it inactivates it. Phosphatase, it dephosphorylates it, but it activates it. So this also, the PDH, it gets deactivated in high energy states such as high ATP, high NADH, or high acetyl-CoA. It also gets activated now with high calcium and in low energy states such as high AMP or high ADP. We got that shit going on. That's all for that, I think. Also, the coenzymes required for this: E1, it requires thiamine pyrophosphate. E2, it requires lipoic acid and NAD and coenzyme A. E3, it requires NADH, NAD, and FAD. And yeah, that's it for that. Now we've got this acetyl-CoA. Acetyl-CoA combines with oxaloacetate. This is the start of the Krebs cycle. It combines with oxaloacetate and then it gets converted into citrate by the enzyme citrate synthase. This step, it releases, it uses H2O. I don't know if that's important, but we got the formation of citrate. The citrate then gets converted into isocitrate. It gets isomerized. The enzyme for this is aconitase. A thing to know about this is in rat poisoning, there's some fluoroacetate, and this inhibits this enzyme, and then it just halts the whole TCA cycle over there. After that, this isocitrate gets converted into alpha-ketoglutarate. We have the isocitrate dehydrogenase. This is also activated by low energy states such as high AMP or calcium, and it's also deactivated by high energy states such as high ATP or high NADH. This step also releases an NADH. Now we've got alpha-ketoglutarate. This then gets converted into succinyl-CoA. Enzyme for this is alpha-ketoglutarate dehydrogenase. This is also a complex like PDH, and it gets activated by low energy states, and it gets deactivated by high energy states, so NADH, high ATP, and it requires the same coenzymes, lipoic acid, NAD, FAD, coenzyme A, thiamine pyrophosphate. Boom, now we're on succinyl-CoA. This also releases another NADH. So we've got 2 NADH formed right now and 2 CO2s formed right now from both the dehydrogenases. Now we've got succinyl-CoA. That gets converted into succinate. This is by this is by thiokinase. Succinate thiokinase, right? Please correct me if I'm wrong. Yes, okay. Succinate thiokinase, yes. And this releases a GTP. Formation of GTP. We've got now succinate. Now succinate gets converted into fumarate. Fumarate, and this is by the enzyme.
Succinyl thiokinase, but Ninja Nerd in his lecture said succinyl-CoA synthetase or something.
This shit about Ninja Nerd, huh? What do you got me, Dr. Crep, looking at you right now?
What are the rate limiting steps of Krebs cycle? Actually,
Krebs
how many
cycle.
are there?
Rate limiting, probably just like what, 1, 2, 3, 3?
Yeah, 3. Which
Yeah.
one?
All the reversible ones, so citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase.
Yep.
Then we've got succinate dehydrogenase. It converts succinate into, is it dehydrogenase? Yes, succinate into fumarate. This releases a FAD. Fuck you, man. And now we've got this FAD. Now, what do you have? Fumarate. Fumarate gets converted into malate by fumarase. This malate gets converted into oxaloacetate again by malate dehydrogenase. This releases the final NADH. So now in total we've got 3 NADH, 1 FADH, 1 GTP, and 2 CO2. So if you try to think about the energy in one cycle, we've got 3 times 3, that's 9. 9 plus 2, that's 11. 11 plus 1, that's 12. 12 in one cycle, but since one glucose makes 2 pyruvates, that means we've got 12 times 2, that's 24. And now if we add the NADH from the link cycle, since we've got 2 NADHs over there, that's 24 plus 6, That's 30. And now if we add the ATP from the glycolysis, that's 6 ATP over there, 36 ATP. Bada boo, bada beam. That's 36 total net ATP, but then in total we have 38 ATP. Oh my goodness. Oh my.
38 in like some other organisms, no?
No, but then it's total net is that वो
looking at the
glycolysis में
looking
2 हम हटा रहे हैं. I think we're done.
I mean, we pretty much are. Alpha ketoglutarate is produced by the oxidative deamination and transamination of amino acid glutamate.
There's some arsenic poisoning over here, right?
Cyclic regulation.
Arsenite, it inhibits which reaction?
Arsenite inhibits
Pyruvate
PDH.
dehydrogenase?
PDH, yeah. Trivalent inhibits
Nucleic acid.
the dehydrogenase complexes, and arsenate inhibits G3P dehydrogenase.
Arsenate and arsenite. Arsenite is inhibiting the dehydrogenase. Okay, we're chilling now. We're done with this. Next one.
Oh, yeah, no.
Gluconeogenesis. How many pages? 1, 6. Oh, this is pretty small too. Chat, are we going for 2 for 2? Are you done with yours?
Let's go.
Like, do you wanna?
Yeah, I'm done.
Done.
I'm
All
listening
right.
to you. Yeah.
127 till ChatGPT this real quick. 133. Boom, 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 boom. 3. What chapter number is this? Chapter 10? Lippincott chapter 10. Boom. Open it up on the other screen now.
Are you uploading this on chat?
What?
GPT पे upload करो.
Yeah.
Damn.
I am going to upload. Wonderful. I'm gonna head to sleep after gluconeogenesis.
After this?
I mean, I'm not sleepy, but I feel like I should sleep. So I can lock in tomorrow.
Honestly, same. But the thing is, all I have left is biochemistry and embryology.
Oh, I've got all of anatomy left.
Okay, you should be worried then. You should be worried, dude.
I mean, bro, gross is not that much, bro. Okay, I feel I could do it tomorrow.
Oh, you can do it. You can 100% do it.
I'm gonna start gross at 12 p.m. Probably finish it by 12 a.m. and then I'll sleep.
Inshallah.
How long did you take for gross?
Another thing with me is that these past couple of days, I just find people and just start talking to them, and I start yapping and wasting my time. But except for that, besides that, I completed it in 1 day from Snell. From Snell, that's the key point there, because Snell doesn't really have all that much.
Okay. Alright, that's pretty good. And how long was that 1 day though?
Like, I stood up till 4 a.m.
And when did you start?
Probably around 2 p.m.
Okay, fair. Well, and you did the hand as well?
Now, the thing I did was, because since I remembered most of the muscles already, like from block one, I remembered a general like idea of the muscle, so I just ditched that. I wasted a lot of time on it, by the way. I like stopped midway because a friend of mine told me like why was I wasting my time on that?
Why? Okay,
And
what did you
then I
do?
moved on to the rest of the stuff: the arteries, the clinicals, the nerves, the joints. I skipped some of the joints, and like two of the joints, but yeah, I did those, and that's pretty much it. I did mostly the clinicals because that's like the most important stuff. And as far as muscles and bones go, I kind of remember those somewhat.
Okay, let's get this shit now.
Lactate from anaerobic glycolysis.
Glucose from non-carbohydrate precursors.
Is Cori cycle in our syllabus? Why do I remember reading Cori cycle? Hold up.
I remember reading it too. It was in the last chapter or somewhere.
No, like I remember reading it as an LO for us to study.
I'm looking to pass the pre-file. I'm not looking to ace.
The same.
But I'm gonna miss on the complete hand region. I'm not gonna study the hand region.
You mean the muscles or what?
Muscles.
I mean, you can kind of ditch the muscles. I mean,
What's important in the hand?
muscles
Just the arches, right? The arches.
and the yeah, the palmar arches, and then there's
The
the
clinicals are just
tibia, but
related
that's
to
not really big.
just the bones.
Oh yeah. Also the facial spaces, you know,
Ah.
the thenar and mid palmar space and the pulp. No, pulp tha kya tha?
I feel like I shouldn't do it because I haven't studied embryo as well. So I'll do like the upper limb until the hand, just generally the hand,
Alright,
and then I'll do the rest
let's go
embryo.
on the clinicals first. Like,
How long did you take
just
for
focus
embryo?
on
Oh,
like.
you didn't do embryo.
No, I haven't done embryo yet. Yeah.
My clinicals were good last time, bro. Clinicals are easy for upper block. Fucking golfers, tennis
Clinicals
elbow,
are the
looking
most
ass,
important thing.
waiter's tip, chip, ape, monkey.
Herbs, palsy, something
Yeah,
else.
bro. Just once you do the what's it called, the brachial plexus, and if you just know where the nerves are present, you can just know like everything. You just gotta know what nerve
Yeah,
supply
brachial plexus.
applies what, and
As
then you
long as
just.
you study the brachial plexus quite
That's
well,
the only thing you gotta do in depth. Maybe the anastomosis and so. All the vasculature and the nerve supply is important.
vasculature, like it's just important for like where the pathways go and stuff, and the anastomosis.
Anastomosis.
Baki the
Also, I remember last time there was some drainage of the lymph nodes and shit. I don't know where that was.
axilla.
Axilla, axilla, and the pectoral
I
drainage.
Yeah, it's not hard honestly. Quite easy.
Uh, let's do this pretty quick though. Got 30 minutes.
Yeah. Let's go.
So gluconeogenesis, it's a synthesis of glucose from non-carbohydrate precursors. So this is done to maintain the plasma glucose during the fasting or starvation states. Yeah.
Mhm.
And this is usually done after depletion of glycogen supplies, which last for around 10 to 18 hours. And then this glucose is synthesized from de novo from lactate, pyruvate, glycerol, and amino acid. Most of the gluconeogenesis occurs inside of the livers, around 90%, and the rest is kidney cortex, which increases during prolonged fasting.
Okay, I have the wrong notes open. What's gluconeogenesis here?
Alright, so the substrate for gluconeogenesis, precursors that make it. First of all, we got glycerol. Glycerol, it gets made from the breakdown of the tags in the adipose tissue during lipolysis.
converted to G3P, glycerol 3-phosphate by glycerol kinase, I think.
Yeah. Oh yeah, there's adipose and liver shit about this.
And then converted to dihydroxyacetone phosphate, which is then
Yeah.
converted to G3P, glycerol D3-phosphate, or something.
Glycerol is phosphorylated by glycerol kinase from glycerol phosphate, which is then oxidized to glycerol phosphate dehydrogenase.
Lactate from anaerobic glycolysis, blood from exercising skeletal muscles, erythrocytes. Lac mitochondria in the Cori cycle, taken up by the liver and oxidized to pyruvate, converted to glucose. I think I have notes on the Cori cycle.
Oh, this is the Cori cycle, bro. It's literally right here in front of our faces. It's easy as shit.
Yeah,
We were thinking all of that.
I got it in more detail.
Okay, okay.
It's not required, but yeah.
Cori cycle describes the exchange of lactate and glucose between the muscle and liver. So now we're in the muscle, glucose gets converted into lactate, gets transported in the blood, goes into the liver, lactate gets converted back into glucose. Gets transported in the blood, goes back to the muscles. Okay, yeah.
Basically, glucose, glycogen is converted into. Actually, ये क्या है? मैं क्या पढ़ा रहा हूँ? मैं क्या बोल रहा हूँ?
Yeah, well, cycling system prevents lactic
Okay, so
acidosis.
glycogen is in the muscles, right? There's glycogen in the muscles, so that's converted to glucose 1-phosphate by glycogen phosphorylase, and then converted to glucose 6-phosphate by mutase. And that can go multiple ways. It can become a pyruvate that is converted into lactic acid and taken to the liver, and जो भी कुछ होना हो वहाँ पे हो जाता है उसके साथ ग्लूकोज में कन्वर्ट करने के लिए. Or it can also be bonded to glutamate to convert it into alanine, and then that's taken to the liver and basically the same thing. It's too complicated, not needed. Pyruvate converted from
Amino acids released from muscle protein breakdown become carbons. This is the carbon skeletons. Alpha keto acids, alpha ketoglutarate, into the TCA cycle, and phenol acetate, precursor of phosphoenol.
pyruvate.
Okay. We got three non-carbohydrate precursors for gluconeogenesis. We got glycerol, we got lactate, and amino acids. Glycerol, it gets released during lipolysis. This is when hormone-sensitive lipase it breaks down. We also got breaks everything down. We got glycerol release. Okay, this glycerol then gets transported into to the liver. Over here we have glycerol kinase, which is not present in adipose tissue. This glycerol kinase phosphorylates glycerol into glycerol phosphate. Then glycerol phosphate gets oxidized by glycerol phosphate dehydrogenase to form dihydroxyacetone phosphate. This is now common between glycolysis and gluconeogenesis. This becomes glucose. Anyways, now next we have lactate. Lactate, this is related to the Cori cycle, in which exercising muscle, it converts glucose into lactate anaerobically. This lactate is transported into the blood, picked up by the liver again, converted back into glucose by gluconeogenesis, which is then transported back into the blood and given to the skeletal skeletal muscles or the exercising tissues. This is the Cori cycle. And lastly, you have amino acids. And amino acids in starvation or prolonged fasting, they get broken down and they form alpha keto acids, which then contribute in the TCA cycle, such as alpha ketoglutarate. The TCA cycle ultimately forms oxaloacetate, which is a direct precursor to phosphoenol pyruvate, which is involved in gluconeogenesis as well. That's that. Only the glucogenic amino acids can do this, not the ketogenic. Now the reactions. Now we had three steps of glycolysis which were irreversible, right? The first one was the first ever reaction, which was glucose kinase, hexokinase, and glucokinase. Those steps were irreversible. That step was irreversible. Next, we had phosphofructokinase, which was irreversible. And then lastly, we had pyruvate kinase, which was irreversible. Phosphofructokinase converted fructose 6-phosphate into fructose 1,6-bisphosphate, and pyruvate kinase converted phosphoenol pyruvate to pyruvate kinase.
The initial step of adding 1-phosphate onto glucose, that was irreversible?
Yes, that is done by hexokinase and glucokinase. That is irreversible. That is irreversible?
regulation पढ़ ली उसकी?
Huh.
Okay, okay.
Tana, I can tell you if you want.
ना 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 मैंने पढ़ ली थी वो
पढ़ ली थी बस चल.
that was the main thing that like stumped me at first कि ये कहाँ से आ गई मैंने कभी पढ़ा नहीं है.
नहीं मुझे याद आ गया था.
Okay, moving on.
I remember this carboxylation of pyruvate.
Irreversible.
Yeah, so they have to be bypassed, and this is done by 4 alternate enzyme reactions. This is basically all we have to know. The 4 reactions. Okay, so now the first roadblock is the conversion. So now we have pyruvate, right? We want to go back. But to go back from pyruvate to phosphoenol pyruvate, that can happen because that's pyruvate kinase, irreversible. We got to bypass this now. So there's 2 reactions which cause this bypassing. This pyruvate gets first converted into oxaloacetate by pyruvate carboxylase. Now pyruvate carboxylase, since it's a carboxylase, it requires biotin, which is vitamin B7, as a coenzyme. Is there a coenzyme or a prosthetic group?
B7.
Yeah, B7. Yes, and this biotin, it gets bound to the lysine residue. I don't think that's important, but that's that. And then it requires biotin.
And uses carbon dioxide to make to carboxylate pyruvate into oxaloacetate. That's pretty much it.
Yeah.
In the mitochondria of liver and kidney cells. It has 2 functions: allow production of phosphoenol pyruvate and to provide oxaloacetate that can replenish the TCA cycle.
Okay. Yo. Chat, this might be
Okay,
low-key easy.
In the presence of cortisol, all of this is amplified. The pyruvate carboxylase and the Pepsi K reactions. I don't know, maybe because stress, you need more glucose. You know,
Yeah.
you need to be more ready, and you need energy for that. So gluconeogenesis, boom, cortisol. Okay, yeah, cortisol stimulates every single enzyme on this pathway.
Okay, now let's just do the first two steps now. Recall. Alright. Since we have three irreversible steps inside of glycolysis. First one being the first ever step, which is requires glucokinase and hexokinase. The next one being requiring phosphofructokinase, which converts fructose 6-phosphate into fructose 1,6-bisphosphate. And lastly, it's pyruvate kinase, which converts phosphoenol pyruvate into pyruvate. Now, gluconeogenesis, if we start with pyruvate, it cannot be turned back into phosphoenol pyruvate because this is irreversible. So then, this gets redirected. The first enzyme is pyruvate carboxylase, and in here we're converting pyruvate into oxaloacetate. Now, this carboxylase, it requires biotin, which is vitamin B7. Yes, and what it does is, and it also requires ATP for energy, ATP hydrolysis. So first of all, this ATP is used to transfer the CO2 to this biotin, and then the biotin, the enzyme, then it transfers the CO2 from biotin to form oxaloacetate. So we got the formation of oxaloacetate. Now we need to move this oxaloacetate out of the mitochondria into the cytosol, but this can't happen because oxaloacetate cannot cross the mitochondrial membrane. So we go back and we convert oxaloacetate back into malate. This was, you can see in the end of the TCA cycle, it was malate to oxaloacetate. So it's just the reverse of that. And since that was forming NADH, this is gonna use NADH, and this is by mitochondrial malate dehydrogenase. Once you have this malate, it can cross the mitochondrial membrane. Now we're in the cytosol. It will get reconverted back into oxaloacetate by cytosolic malate dehydrogenase, and this forms the NADH again. Now this oxaloacetate gets converted into phosphoenol pyruvate by PEP carboxykinase. And the energy for this one, instead of ATP this time, it's by GTP, and this releases CO2. So before we use CO2, and now we're releasing this CO2.
Yeah. And another thing regarding the movement of malate outside of the mitochondria, there's a usage of an NAD to
Yeah.
take malate outside, to convert it into malate. So it also helps in the transport of malate. This is where the malate aspartate shuttle comes in for the
Oh.
transport of malate, NADH, inside and outside the cell, the mitochondria.
Let's see if there's like a photo for this shit. Malate aspartate shuttle.
Oh, there are 100 photos.
Just memorize the freaking photo.
Try and look at
Malate.
this. Yeah, this one.
Malate. Aspartate. Oh, what? Is it that simple?
I mean, I feel like you shouldn't really know more detail than that.
Okay, wait. Where the fuck is the Nitazole matrix. Okay, this is the matrix. We've got malate. Huh? Wouldn't it be the opposite? Yeah, I'm not tripping.
The thing is, it's used to transport NADH from outside of the mitochondria inside the mitochondria. So oxaloacetate, so malate, it's transported in. Well, malate की formation कैसे होती है? Malate is formed by the reduction of oxaloacetate into malate. So 1-NADH is used to form malate, and then that's transported into the mitochondria, and then that's used to convert it into then oxaloacetate and then form an NAD, I think.
So this is what to transport NADH into the mitochondria.
Yeah, just into
Mainly.
the mitochondria. That's
Oh,
mainly.
okay. This is quite simple. Mitochondrial malate dehydrogenase. Okay, and then aspartate aminotransferase.
Yeah, aspartate is just like used to transport the molecule outside, I think, to then again be used
Because
as
the
a
oxaloacetate can go out.
NADH carrier.
Aspartate aminotransferase. Okay.
Yeah.
It converts. Okay, that's quite simple, bro. We just did this in 2 seconds. No way we're stressing about that shit. Also, another thing. This oxaloacetate, it can also be converted directly into phosphoryl pyruvate inside of the mitochondria and just transported outside. That's another thing that can just happen and bypass all of this bullshit.
Huh?
Yeah.
Yeah. No,
So we don't gotta do these fancy tricks doing this shit.
pyruvate transport into the mitochondria, carboxylated by PC to OA, which can be converted to P by the mitochondrial enzyme, and then transported into the cytosol. What the fuck?
Anyways, now the rest of the reactions, they occur the same as glycolysis until we reach fructose 1,6-bisphosphate. No. Phosphorus is one acute reversible fructose 1,6-bisphosphatase. Huh? Is it that simple, the name? Okay. I don't mind that.
Phosphofructokinase कौन सा वाला? ये वो wait मुझे कुछ समझ नहीं आता.
Oh, yeah, it makes sense. Hold on. Chat. Now, okay, we did the first two. You understand the first two steps, right?
Yeah.
Now we're going back up, right? So now we reached fructose 1,6-bisphosphate. तो उसमें जो रिएक्शन था fructose 6-phosphate to fructose 1,6-bisphosphate, that was irreversible क्योंकि हमारे पास था fructose phosphofructokinase 1, right? And you remember how phosphofructokinase 1, its main activator was phosphofructokinase 2.
Yes.
You remember that, right? और फिर ये बायफंक्शनल एंजाइम था, उसका दूसरा पार्ट कौन सा था? फ्रुक्टोज बिसफॉस्फेटेज 2. और वो क्या करता था? फ्रुक्टोज बिसफॉस्फेटेज 2, it converted fructose 2,6 into
2,6-bisphosphatase.
Yeah, 2,6-bisphosphatase. It converted fructose into so since this is 2 and now we're talking about 1 right now, all we gotta do is make it fructose 1,6-bisphosphatase. Chat, this makes so much sense. Oh my days. This is perfect. This is like, you know, the
Also,
puzzle piece is just
fructose 2,6-bisphosphate Like back when we were seeing it, it was a strong activator, stimulant to the whole glycolysis reaction. It also works to inhibit gluconeogenesis by inhibiting the enzyme that helps in the reverse reaction.
Yeah, okay. That makes even
So
more
it
sense.
helps in the regulation of those two.
This is wonderful. Low-key made me
Uh-huh.
kind of happy. Location cytosol, mainly in the liver and kidney. This is the regulatory control site of gluconeogenesis. That's all I hear. On my days, I'm on 11 hours now. Right now. Rien serious right now, bro. Have you done physio?
That's my long. Physio. Yeah, I'm done with physio.
That's fine. Alright.
I didn't do physio from Guyton. I have like, you know about Pocket Guyton, right?
Yeah.
So basically, I just read through Pocket Guyton, and by the way, this took me 2 days. That's how much I was procrastinating all of this stuff.
Oh my goodness.
I read through Pocket Guyton in 2 days,
I mean, I
and
started
half of
studying
it
yesterday too. I finished physio, half of physio yesterday, half of physio today, and half of biochem today, and I guess it's still today, so half of biochem today, and then all of anatomy today. Half of anatomy today and half of anatomy tomorrow, you could say. We'll learn all nighter for this one. Wonderful.
Okay, let's get through this. Let's get
Yeah.
through this. Let's read this.
Cytosol, mainly liver and kidney, regulatory control site. Inhibited by low energy versus gluconeogenesis. Huh?
What page are you on? Oh, this one. Never mind. This is converted to that by once is inhibited by that very strong inhibitor. So cortisol, of course, cortisol. हर जगह देखो cortisol.
Hmm. So this could be regulation. It's the same thing. Okay, let's flip it. Flip the freaking.
Where's AMP? AMP is also an inhibitor of this friends, isn't it?
AMP's inhibitor, yeah. बस जो भी था हमारा फॉस्फोफ्रुक्टोकाइनेज का जो भी उसके रेगुलेटर से it's literally the complete opposite.
If there's high AMP, then it's gonna get inhibited. If there's high ATP, it's gonna get
Regulator.
जो भी है.
Okay, now we just have one more thing to bypass, and that's the glucose 6
Glucose
phosphatase.
6-phosphate. Glucose 6-phosphatase
ठीक है.
present in the smooth endoplasmic reticulum of the पता नहीं क्या smooth endoplasmic reticulum में होता
Smooth endoplasmic reticulum. Luminal surface of the endoplasmic reticulum. Is it smooth? I don't know about smooth. It's just the endoplasmic reticulum. I don't know if it's
है.
smooth or rough though.
I have smooth endoplasmic reticulum written
Written?
here.
Okay, in the books then.
Not on Lippincott, not on Lippincott. I got notes.
Oh no, no, screw it then. I'll just do endoplasmic reticulum.
Just endoplasmic reticulum. It's
Yep.
also a biomarker for the endoplasmic reticulum.
Oh yeah,
That one.
glucose 6. Glucose 6 phosphatase is on the endoplasmic reticulum, then converts glucose 6 phosphate into glucose. It probably releases phosphate as well in the process. Yeah.
And then we got glucose.
Only liver and kidney possess this enzyme. Can release freely glucose into the blood. Glucose 6
And then
fast.
you got
Oh, there's two. Requires two proteins: glucose 6 phosphate translocase and phosphatase. Translocase it transports it to the ER, and the phosphatase does the phosphatase job. Muscles lack it, so its glycogen
where's
cannot maintain blood glucose.
either एक second. Um where's the translocase? Okay, yeah, you have translocase transports it inside, then removes that. The endoplasmic reticulum for proteins are also required for the final step of glycogen degeneration. Oh, I see. And then you got glycogen storage diseases caused by deficiency of the phosphatase and translocase.
Yeah, I'll do that tomorrow. I think I just have glycogen metabolism and
Written in the same paragraph.
yeah, it's the type 1 glycogen storage.
Because free glucose is unable to Mm, okay, this makes sense. Makes sense. Makes complete sense. Yes.
Yes, yeah.
Very good.
Very good, very good. 2 ATP into 2 ADP.
Wunderbar.
So we were using up 4 ATP, 2 GTPs. Oh my goodness. Are you serious right now? Using up 2 NADHs. We're using up 4 ATPs and we're using up 2 GTPs and we're using up 2 NADHs. Oh my goodness. What an inefficient process.
Okay, so stressing actually helps you burn more calories
Why?
because cortisol causes gluconeogenesis. It's like one of the main things that like it's kind of an important point where
Okay.
you stress, there's cortisol. Cortisol causes um increased activity of all these enzymes.
Okay. Glucagon decreases hepatic fructose 2,6 bisphosphate. Yes. Inhibits phosphorus 1, slows glycolysis, and enhances gluconeogenesis. Glucose production. Glucagon binds to G protein coupled receptor in hepatocytes. That causes the GDP to be replaced by GTP, which causes the alpha subunit to dissociate, activating adenylyl cyclase, which converts the ATP into cyclic AMP. Cyclic AMP then causes the formation of protein kinase A, activation of protein kinase A. That then causes phosphorylation of whatever.
The who, the
Good.
what now?
The who, the what now?
Oh, this hormones acting on PA for protein kinase A, phosphorylation of the. Oh, I see, I see. ये तो वही ग्लाइको वो क्या नाम है उसका what was it called glycolysis वाला point है इसी हद तक.
PEP is diverted from pyruvate formation towards glucose. PEP is diverted from glycogen from pyruvate formation towards. You induction of enzyme synthesis. Glucagon increases the transcription of PEP carboxykinase. Oh wow.
ये तो बस मतलब general point है कि insulin causes increased activity and increased transcription of all the enzymes that's associated with it and decreases the ones that aren't associated with it.
Yeah.
Glucagon opposite. Carbohydrate response
Low
elevated.
insulin promotes proteolysis. Gene fasting. Low insulin.
Cortisol also increases the gene expression.
ATP. Fatty acid. Oh my god, how am I gonna study block 2 in 2 days? In 1.5 day. Shoot.
Wait, you have to study block 2?
I haven't studied block 2 or block 3.
Like, oh, I see, I see, I see.
No, like deadass, I haven't studied it.
मैंने block 3 का एक लफ्ज़ नहीं पढ़ा. And for block 2, I haven't studied biochem. Embryology तो मैंने university नहीं पढ़ी सबके लिए.
And block 2 biochem is very easy.
Yeah, that's what I'm counting on, honestly.
Very easy. You could do it in what 6 hours, bro.
Yeah, but I want to spend those 6 hours for embryo tomorrow. If it takes me 6 hours, which I know it's gonna take me.
Block 2 का block 1 के बाद पढ़ो ना.
मतलब वही ना वही करूँगा. I'm talking about block 1
Mm.
embryology instead of block 2 biochem tomorrow.
Yeah, yeah. Steric activation. Block still. Use life policy. Okay, we're done. Done with this chapter.
He's done, boys. He's done, boys. How much am I left to? I'm left with like one page. Mm. This was the third chapter, right?
Yes.
Okay, so after this, there's the glycogen chapter.
This is the fourth chapter. There's an introduction to as well. इसके बाद glycogen है और pentose phosphate pathway. HMP short.
HMP shunt, yeah.
That and then I'm not gonna do gags. I'll just do the. I didn't do the fructosuria thing last time. Do with that.
No, I'm gonna do that. It's quite a short chapter, honestly, and it's
Yeah, I'll
easy
do
to
it.
understand.
Yeah, Rana.
It's not complicated.
3, 4, 5. Do it tomorrow. So I'm gonna sleep for 4.5 hours. That's around 3 sleep cycles.
Insane.
Let's see, for 3 sleep cycles. Gonna wake up if I sleep around 3:30 today, and I wake up at 8, and I start studying at 8:30. By 12, I should be finished with biochem.
By 12, you should be finished. Mm.
And then all I gotta do is some anatomy. And low key, if I finish gross anatomy by 6 p.m., we're gonna do some embryo.
Gross it is.
If I finish gross anatomy by 6 p.m., this is bold claims right here. Bold claims.
Low key chilling.
Chat, thank you for all the support today. 11 hours and 12 minutes of studying today. Probably 2 hours of breaks in between, but nonetheless, wonderful. Wonderful indeed. We're gonna start the stream again in around 5 to 6 hours. See you guys tomorrow.
Yeah, cue the outro.
Cue the outro, chat. Cue the outro. Oh my goodness, my back's tired.